For as long as humans have cultivated land, they have fought an invisible war, not against insects, not against drought, but against competition. Weeds compete for sunlight. They draw water from the same soil. They absorb the same nutrients crops depend on. Even small infestations can quietly reduce entire harvests. For thousands of years, farmers fought back with force. They pulled weeds by hand. They tore roots from soil. They turned fields with steel blades and plows. They burned growth back to ash. It was exhausting. It disrupted the earth itself. And the cycle never stopped. Because weeds don't just grow on the surface. They grow through biology. In the 20th century, agricultural chemistry introduced a different idea. Instead of destroying plants physically, what if you could interrupt their internal machinery? That idea led to glyphosate. To understand how it works, we have to zoom past the field, past the leaf, and into the chemistry inside a plant cell. Plants build themselves from amino acids. These molecules assemble into proteins. Proteins shape structure. They regulate growth. They power photosynthesis. They control cell division. Without amino acids, a plant cannot continue building itself. Among the pathways that produce these amino acids is something called the shikimate pathway. It exists in plants, in fungi, in many bacteria, but not in animals. That difference matters. The shikimate pathway produces 3 essential aromatic amino acids: phenylalanine, tyrosine, tryptophan. These are not optional components. They are required for protein synthesis, for metabolic balance, for survival. At the center of this pathway is a single enzyme, EPSPS. Think of it as a gatekeeper in a molecular assembly line. Without it, production stops. Glyphosate is designed to bind to that enzyme. When it attaches, it blocks EPSPS from functioning. The pathway slows. Amino acid production halts. Protein synthesis begins to fail. The plant does not burn. It does not collapse instantly. Instead, it experiences metabolic shutdown, a slow biochemical interruption. Over time, growth stops entirely. This precision, targeting one enzyme in one pathway, is what made glyphosate significant. Because animals lack the shikimate pathway, the biochemical target exists only in plants and certain microorganisms. That selectivity shaped how glyphosate was used in agriculture. In the 1990s, biotechnology introduced crops engineered to tolerate glyphosate exposure. These crops contained a modified version of the EPSPS enzyme. Glyphosate could no longer bind effectively. Farmers could spray entire fields. Weeds were suppressed. Crops remained intact. This reduced the need for mechanical tilling. And that mattered. Tilling disrupts soil layers. It increases erosion. It releases stored carbon into the atmosphere. Reducing tillage helped preserve soil structure and moisture retention in many systems. Glyphosate became widely used not because it was dramatic, but because it was efficient. It targeted plant metabolism with specificity. But biology responds to pressure. Over time, some weed species evolved resistance. Through natural selection, plants with slight genetic variations survived exposure. Those survivors reproduced. Resistance is not unique to herbicides. It is a pattern seen wherever selective pressure exists. Antibiotics, pesticides, evolution itself. The story of glyphosate is not a simple one. It is a story of molecular interaction, of agricultural engineering, of biological adaptation. At its core, it is chemistry interacting with life. One molecule, one enzyme, one pathway, but the effects ripple outward, from plant cells to farming systems to global food production. Chemistry does not argue. It binds, it blocks, it reacts. And sometimes, at the smallest scale imaginable, those reactions reshape entire industries,